Thank you, Tsu, and good morning. My name is Rachele Sangaletti. I'm going to be the moderator for this second session, Emerging Tools and Techniques in Neuroscience. We have 6 outstanding speakers for this session, and I would like to thank you, all of them, for accepting our invitation to the Neural Engineering Symposium. Just a reminder for the speakers, you will have 10 minutes to go through your presentation. And after this section, I will read the questions for you all. So we can start with our first speaker, Dr. Rene Barro. Dr. Barro is a research assistant professor at the Department of Medicine at the University of Miami. And today he will talk about voltage sensing mechanisms of neural K, C, and Q channels. Go ahead, Rene.
Thank you very much, Raquel, and thank you very much the organizers for this great symposium. So today I'm going to be talking about voltage sensing mechanisms in potassium channel that is expressed in neurons. So I work in the Department of Medicine. My name is Rene Barro Soria. So the outline of the talk, it's really simple. So first I will give you a perspective of the physiological and pathophysiological roles of these channels. Then I will talk a little bit about the properties of K-Q channels. Then I will introduce you to the main technique that we use to study the structure and function of this channel. And if I have time, I will give you a quick overview of how we use this technique, this powerful technique to actually understand how disease associated mutation, in this case epilepsy is our main focus, leads to channel dysfunction and causes epilepsy. So let's start with the physiological and pathophysiological roles. So these channels in health, so these channels are really expressed throughout the system, I mean the body, and they comprise 5 members, the family, so K-Q1 or KV 7.1. So I will be talking about K-Q, but it's also KV 7, 1, and 5, and 1 it is expressed in the heart, whereas 2 and 3, my main focus, are expressed in neurons. 4 is expressed in the inner ear, and 5 in the muscles, and so forth. But these channels are also important in disease because mutations in either of these channels causes a severe disorder, such as, for example, it causes life-threatening cardiac arrhythmias, deafness, it causes neuropathic pain, also epilepsy, as I mentioned, and autism spectrum disorders. So there is really an ongoing need to understand how these channels work to just tackle the different diseases that they are involved in. And that is so because we need to, in understanding how the channel work, we actually will be able to develop effective therapies. So for example, in the case of epilepsy, just to put one of these disease that we are interested in, this is a disease, this is a chronic disorder that affects people of all ages. And we are talking about 50 and 60 million people in the world. That makes one of the most common neurological disorder in the world. And interestingly is that 30% of patients suffering from epilepsy are refractory to existing drugs. So they don't respond to drug really. So we are, studying these channels to just develop small molecules that tackle the channels. So as you know, excitable cells communicate by electrical and chemical signals. So here I show you a representation of an action potential of a neuron. And as you know, the rise phase here is really developed through an inward current of inward sodium current generated by voltage-gated sodium channel. And the repolarization of action potential is due to outward potassium current through voltage-gated potassium channels. However, the channel that I'm going to be talking about today is a channel that is none of these channels, but it's a channel that is really active in the negative voltage range here. And therefore, this channel has been implicated in actually establishing or helping establishing resting memory potential and impeding repetitive neuronal firing. So this is a current, we call it GKV7 or GQK channel, opposes this inward sodium current. So this is an outward current that actually happens here at a threshold, around threshold of action potential. So here, this is a quick example to show you why these channels are so important, and they work really as a brake on repetitive neuronal fire. So if you take a rat sympathetic neuron, for example, here, and you measure in a current clamp mode, you measure action potential, you see that it generates only one action potential and then is silenced. And this is because these channels represented here in a voltage clamp mode, it's open. So when the channel is open, this efflux of potassium, positive charge out of the cell, really silences the neurons. And when the channel is blocked, as you see here, or in the way to be blocked, then you have more of this action potential occurring. So that's why these channels, patch clamp experiment and so forth have been really showing that this channel function as a brake on repetitive undesired action potential. So the research goal in my lab actually are to understand how these channels function, so that we can understand the mechanisms by which mutations lead to the functional channel. We call it that channelopathies. And then all of this with a real aim to design drugs that correct these channelopathies. So to just give you an overview of the properties of this channel, these are voltage-gated potassium channel, the casing Q, that forms tetramers with very distinctive voltage sensing and pore domain. So here I show you one of these subunit, one of the four subunits. 1 to 5 transmembrane, 1 to 4 transmembrane domain forms the voltage sensing domain, and 5 and 6 forms the pore. And as you can see, well, 6 is the gate, and as you can see here, the voltage sensor in red has several positively charged residues that actually work as the voltage sensing structure. I show you here a cartoon representing, the pore aligning of 5 and 6 central pore, surrounded by 4 of these voltage sensor, if 4 of these subunits come together. And this is just a top view of a recently a structure of casing Q1 with its 4 adjacent voltage sensor domains, you see here, and the pore in the center. So here I show you also an example of homomeric and heteromeric casing Q channels. Casing Q2 homomers, casing Q3, and when we combine casing Q2 and casing Q3, which is really the M current in neurons. So a combination of casing Q2 and casing Q3 represented here by blue and red. So when 2 of either subunit come together, that forms the M current. And as I mentioned to you before, a lot of mutation in the meantime has been identified in both channels that causes severe diseases. But I want you to draw attention to the voltage sensor here, S4, that a lot of de novo mutation, it's continuously, being popping up, which shows severe epileptic encephalopathy. So we are really interested in understanding why these mutation really affect the channel. And this is a voltage gated channel. So anything that affects the voltage sensor would affect the overall channel structure. So how do we do that? So that brings me to the technique that I wanted to introduce, the voltage clamp fluorometry technique that we use to study this rearrangement of voltage sensing and a channel opening. So here I show you a cartoon representing how do we study that. So in a cartoon, so we have here the channel represented in a closed state at minus 80 millivolts when the voltage sensor are really down and the gate is closed. And then when we depolarize the channel, this depolarization repels the charges of the voltage sensor. This move up and then opens up the gate to allow ion conductance. But what seems really easy here in this cartoon is it's technically challenging. So it's hard to measure these distances. And as I said before, we have overcome this difficulty by using voltage clamp fluorometry, which is a fluorescence-based assay of voltage sensor movement. So all we do is we engineer a cysteine here in the extracellular loop of the channel. And then we express this engineered cysteine in xenopus oocyte. We wait a couple of days for expression, and then we label with a malamide fluorophore. In this case, we use Alexa, but you could use whatever fluorophore desire. And then we put it in a voltage clamp fluorometry setup to simultaneously measure current and fluorescence. So it will give us information about how the channel, the conformational changes that occurs in the voltage sensor and in the gate with the current. So this is a very simple technique. Actually, the principle is that here I show you a cartoon of the channel in the closed state with a voltage sensor in its down position or the resting position when the channel is closed. And then you see the fluorophore attached to the cysteine. And when you depolarize, the fluorophore moves along with the voltage sensor to a different environment and changes. This changing environment actually changes the fluorescent signal from here to here. And that's what we measure in our system. So it's very simple. And the same is true when we hyperpolarize, the fluorophore again moves down because the voltage sensor moved down. And then this change in environment will be also sensed by our system. So if I now show you an example of, for example, case in Q3, this is the current, how it looks. If we apply this family of voltages of this channel, we have this current, and if we measure fluorescence of the channel, we see nothing. However, when we have the cysteine engineered and we put the fluorophore, we label it with the fluorophore, we are able in the meantime to record fluorescence changes, voltage-dependent fluorescence changes, signaling both case in Q2 and case in Q3. You have here the changes for case in Q2 and the changes for case in Q3. So with this in hand, we have case in Q2, a channel that can, we can study the rearrangement of the voltage sensor and apply this to the current of the channel, and then do biophysical studies to just dissect, the voltage dependency, the kinetics of the channels, and so many other things to study how the channel, the wild-type channel, work, and then with that, understand how mutation works. For example, you can imagine that we can now have mutations in either case in Q2 or case in Q3, and then study the voltage sensor movement and see how that correlates, or heteromerization of these channels, when we can see the relevance of the voltage sensor movement of one subunit, how that affect the other one, and so forth, and many other combinations with the mutations. So I just wanted to give you, if I still have a couple of minutes, Raquel?
You already ran out of time, but if it's only 1 minute, you can go ahead.
Okay, so it's just an example of different epilepsy-associated mutations that we, in the meantime, are characterizing and studying, as you see here. These are mutations in the voltage sensing domain, and we, in the meantime, are very good knowing what's happening with this mutation at the molecular level. And I can tell you that in this case, for example, this mutation that people thought that retains the voltage sensor in its up configuration, in a physiological voltage range, people thought that it was locked and there was no fluorescent movement. But when we use our technique and then we push the cells really, and this channel really to negative potential, we saw that the channel closed and the voltage sensor really happens, as you see here. So the only thing that happens is that there is a shift to very negative potential of this channel. So with this technique, we have a very powerful tool in hand to just dissect how these channelopathies really work. And as I mentioned to you, we not only have mutations in the voltage sensor, but also in other part of the channel. And we use another technique that I will leave it for the private question, but we can, as you can imagine, we can now start tackling different part of conformational changes that occurs not only in the voltage sensor, but differently in other part of the channel with a new technique that is called encoding of fluorescence amino acid, that I will leave you for the question session. With that, I want to thank the lab that has been working on that, and our collaborator, and of course the NIH for funding. Thank you.
Thank you, Dr. Barro. Thank you. Our next speaker is Dr. Khizroev, professor of electrical and computer engineering at the University of Miami. And he will talk about wireless brain-machine interference with electrical nanoparticles. So, Dr. Khizroev.
So my name is Sakharat Khizroev. I'll talk about the wireless brain-machine interface with the magnetic nanoparticles. It's a joint effort, research done during the previous year involving University of Miami researchers. This is the pictures you see of my team, graduate students and postdocs, fantastic guys, and mostly researchers from California, Irvine, CNMI, led by Dr. Ping Liang in collaboration with Neuroscience Professor Ian Smith at UC Irvine and Professor Xiaoming Jin at Indiana University, Purdue University, Indianapolis, also neuroscience department. So outline of my talk. First, I will make an introduction, talk about magnetoelectric nanoparticles. That's the core of the technology, and I will talk our first single neuron level stimulation of the central nervous system wirelessly. Talk about the wireless recording experiments, first recording experiments as well, and conclude. So this introduction, as you all know, I'm sure, most current technologies to stimulate or record neurons today are not wireless. They are quite invasive. So it means they need electrodes, microelectrodes. And you may remember in the most recent, very highly advertised demonstration of by Neuralink, they used approximately 1,500 electrodes. And you can see that 1,500 is actually zero, considering in the human brain there are 80 billion neurons. And the reason is that's why with this technology, no way we can actually understand how neurons communicate. We want to understand how neurons actually communicate with each other and how to control that communication, that stimulation. So with that technology, you cannot do it. So you need wireless, something wireless. With electrodes, you cannot stick 80 billion electrodes in the brain, definitely. So there are wireless technologies, by the way. To write, you probably heard of transcranial magnetic stimulation. The problem is transcranial magnetic stimulation is extremely inefficient. So spatial resolution, even though they claim millimeters, it's actually centimeters, because they put sources outside the brain and they rely on solving that inverse problem to stimulate the actual brain. And it's inefficient because they use fast changing magnetic fields to induce eddy currents. So it's not direct interaction with electric fields. There is wireless recording techniques also exist. Probably the most established today is magnetoencephalography. Again, the same problem, the fundamental problem of mathematical inverse solutions. And they rely on detecting tiny magnetic fields coming from the brain, tens of femtotesla. It's nothing. So they need very expensive, bulky, low temperature cryogenic equipment, and the resolution there is very questionable. So that's the reason. That's what motivates us. We want to do something wirelessly at a single neuron level. So we will be able to reach signal wirelessly from each neuron and also write into this, write and stimulate. So we propose technology based on magnetoelectric nanoparticles. Magnetoelectric nanoparticles been around for a long time, for at least 20 years, but they became popular only relatively recently, a few years ago, and the reason they became popular only recently, they learned to make them work at room temperature. And it's a quantum mechanical effect, and probably the most popular implementation today is so-called core shell structure, where nanoparticles made of two phases: the core, magnetostrictive core, and the dielectric shell. And these two components are strongly coupled, lattice match coupled because of the crystallographic nature, right at the interface, and that's the key. So to write information with this particle, all you have to do is apply magnetic fields. Magnetic fields easily go through the brain. As you know, electric fields do not, you cannot use electric fields wirelessly. That's the reason they use electrodes, because electric fields interfere with very complex circuitry of the electric brain. But magnetic fields do not, but magnetic fields cannot directly interact. That's a problem there. So magnetoelectric nanoparticles allow you to couple these two advantages of these two fields together while mitigating their disadvantages. So what it means, you can use magnetic fields to induce electric fields locally, right close to the nanoparticles. The size of the nanoparticles varies from 10 to about 50 nanometers. So you can bring them very close to the membrane and control action potential, induce action potentials if necessary, using magnetic fields. That's ideal. That's been a hypothesis for a few years, and this is kind of the year we made a demonstration again, as a single neural level. It's easy to say, but engineering, they're extremely complex. The whole thing depends on the ability to generate that magnetic electric coefficient. It's a quantum physics there, guys. It's beyond the scope of this seminar. I'm not going to get into this. But, finally, we managed to do that. And we have talented students, Alex Zhang and Ping Wang, whom, for the first time, they made the structure. For recording, it's a principle of reciprocity. So what you do, every time neuron fires, there is electric field fluctuations around the neuron. So because of the magneto-electric effect, that induces magnetic fields, which are relatively straightforwardly picked up by existing techniques. Again, I apologize, it's not that easy as I told. I take it back, but it can be done. So that's what I'm going to talk about. This is the structure I was talking about. Again, took us 10 years to come to this point, guys. So as you can see, the whole idea is the piezoelectric shell in this particular structure is barium titanite, is connected to the magnetostrictive core. It's one of the ferrites, like oxide material here. In this particular case, we use cobalt ferrite. Today there are tons of different materials, but this is the pioneering material used in this study. What's important to keep in mind, it's not just this magnetoelectricity. For the magnetoelectricity to work, you have to make sure these particles, especially the magnetic core, does not become super paramagnetic. Super paramagnetic means, you see that hysteresis loop, M versus H. It means you cannot have hysteresis-free system. You have to have hysteresis. Otherwise, magnetoelectricity doesn't exist. That's a challenge, to make them small enough and keep that loop there. That's actually that's why it took us several years, and that's how we decided to use high anisotropy materials. Again, I'm not going to talk about it. So actual demonstration. So now, to really stimulate one neuron, selected neuron, any neuron in the brain, for example. Of course, these experiments were done in vivo in Indiana Neuroscience Institute, and in California in vitro, and in Miami using our particles. This particular experiment I'm going to discuss is probably the most obvious ones for the illustration purpose. Was done at CNMI in Irvine. With the University of California, Irvine, by our collaborators. So what you can see here, you can see calcium imaging, 3 figures, showing 3 stages of application of magnetic field profiles. You can see the field of view about 300 by 300 micrometers. You see a few neurons, and we label them, neuron 4, neuron 5. These are 2 key neurons, and our goal is to change magnetic field profiles, spatiotemporal patterns, so we can stimulate first one neuron and then the other neuron. So first we stimulate neuron 4. That's how the field pattern was chosen. So we use external magnetic fields to do that. And in next figure, I'll show you actual the stimulated, activated waveforms. Then what we do, we move using special technology called soft core, magnetic core technology, which creates very large magnetic field gradients, but relatively small fields. We change spatiotemporal pattern of magnetic fields, so that in 2 moves we do. First, you can see the brightness of neuron 4 got low a little bit. What we did, we moved our field center of the field to between these neurons. And in the 3rd stage, right here, figure 3, you see you move. I have to eliminate. I have to get
Dr. Khizroev, you have 2 minutes left.
Okay,
Sorry.
great. Yeah. So then you move to neuron 5, and now you stimulate neuron 5. Okay, going to the next one. And here you can see what exactly is happening, guys. Look, when you see a blue line, we turn on our field. We activate our external fields. So you see, when we turn it on, first we stimulated. We pick the parameters so that our neuron 4 was activated. You see, there is a waveform here. It's about 10 hertz. This is a frequency with which we are sending our signals. And you can see it's perfectly coincides with that. Then we turn it off. That's it. Neuron 4 is off. Then we turn it on again. On. So we can do it infinitely. So it's accuracy is 100%. Reproducibility is incredible. Done on hundreds of dishes. Then we move our field pattern, but you see neuron 5 is not activated. So then we do the same thing with neuron 5. Guys, these neurons are about 80 micrometer close from each other. They're really close. You understand this? We didn't even expect this system would work that well. You can see, and so now we're controlling the next one. So that type of stuff. And the next slide, what you can see that signals, actually activated neurons signals, their amplitude coincide. They are on the same order as a magnitude of activation, calcium ion channel coming from a neuron. It was some random neuron, number 28, which was spontaneously activated. So comparable magnitude and so on. It's definitely activated thing. But we're absolutely confident in this stuff. So again, this is statistics. I know I'm almost running out of time, but again, by the principle of reciprocity, recording never been done wirelessly, guys. What MEG does is I would not call recording. It's very high level, very primitive thing. So the idea, if you can record wirelessly with no wires from such small regions, is almost like sci-fi today. That's why we were excited that we could do that. Yes, so I know I'm running out of time, and I'm not going to describe the physics, but you can see here the signals were recorded by the physics I was just describing. They coincided with what we recorded with our baseline experiment using calcium channel. You see in this figure on the left. This, by the way, these pulses, these are our pulses, recording our particles. This is a calcium image channel thing. They don't exactly coincide, but we definitely can see the same pattern all the time. And our sensor is about 1 centimeter away from a cell culture. By the way, when we did writing, we used cortex cell cultures, and we did recording. We used hippocampus cell cultures for the reason because they are more active. They're easier to keep them active. I'm gonna skip the physics. In conclusion, so I want to acknowledge support from DARPA grant, from National Science Foundation grant, and Air Force Office of Scientific Research, guys. That's it. That's kind of.
Thank you, Dr. Khizroev. Thank you very much. So our next speaker is Dr. Ali, Research Assistant Professor at the Department of Neurological Surgery, University of Miami. And he will talk about integrated high throughput technologies in biology, chemistry, and computer science to discover therapeutics for CNS injuries. Dr. Ali, go ahead, please.
All right. Good morning, everyone. My name is Hassan. I'm a biochemist by training. My specialty is early stage drug discovery, and I'm an assistant professor at the Department of Neurosurgery. One of the things I do at the Miami project is work on the problem of regeneration, nerve regeneration in the central nervous system. And I do this with 2 neuroscientists you've already heard about today, Vance Lemon and John Bixby. And the problem here is that the axons, the processes of the neurons that project from the brain through the spinal cord to connect the brain to the rest of the body, if they sustain an injury and they become severed, they do not regenerate, and that results in permanent losses of connectivity and therefore loss of sensory and motor functions, aka paralysis. And this is believed to occur due to 2 main mechanisms. As our neurons age, they lose their capacity to regenerate. So this is referred to as the intrinsic regenerative potential. Also, there's molecules in the central nervous system milieu that signal to these axons not to grow. So this is referred to as the extrinsic growth suppressive mechanism. So the problem is complicated, and therapeutics that have attempted to treat one mechanism or the other have not been clinically successful. And it's extremely difficult trying to deconstruct a biological process and finding a gene product that you can develop a drug towards. Hard enough to do this for one process, let alone for 2, and then developing a drug that can address both at the same time is extremely complicated. So one alternative approach you could do is test things empirically. And rather than try to deconstruct the system, just test different things on it and see what works. But of course, empirical testing can be time consuming and expensive. So we have made use of several high throughput technologies to kind of overcome this logistic challenge. So one of these things that we use is called phenotypic screening. So rather than deconstruct the neuron and try to figure out all the details of its molecular workings, we take these neurons out of the biological system, out of the body, and we culture them in vitro. And we test things on them directly, and simply we find things that work. And that gets us out of having to understand everything about the neuron before we can find things that can make their axons grow. So, this is what these neurons look like. We take them out, we put them in small petri dishes, and after 2 days, you see that they start growing these processes or axons. And then we image those cultures and we digitize them, and we have software that automatically analyzes those images. So you can see the software can find where the cell bodies are, and it can trace these processes and calculate the length of these for each cell. So from every cell, we can get hundreds of different parameters on the shape and size of the cell nucleus and the cell body and the processes and so on. And this is what the culture looks like when we have an agent that promotes outgrowth. So you can see that the assay is robust, and you can really identify things that promote neuroid outgrowth. In order to save on time and reagents, we miniaturize these assays, and we run them in these microplates. Well, you have small wells. We place neurons in each one of those wells, and then this way, in each different well, we can test a different reagent. So in one of these plates, the one that I'm showing you here, we're basically doing 96 different experiments, and that really cuts down on the cost and increases our throughput. So of course, if we were to test, if we were to image and analyze each one of these wells manually, it will take us a very long time. And this is where the second technology comes in, which is robotics and automation and microscopy. So what we do is we do these tens or hundreds of plates. We fix them after the experiment is over, and we stain them, and then we just stack them up in this room where we have this microscope and this robotic arm. And basically the robot works overnight, just analyzing these plates one after the other, and then the software traces them, dumps the data in a server, and then we just come the next day and analyze it. So this efficiency in the pipeline and the throughput of doing the experimental work needed to be matched with throughput and efficiency in acquiring things to test in this system. So for that, we make use of high throughput chemistry, specifically massively parallel chemical synthesis. And this is just a fancy way of saying, chemists can generate hundreds of thousands or millions of molecules in a single mixture by doing clever synthetic techniques. So around a single scaffold, they can build hundreds of thousands or millions of diverse molecules. So I'm just showing a mock example here of two mixtures, one where the scaffold is represented by a triangle and one by a circle. And the different colors of the nodes represent different chemical moieties attached to the scaffold. And you can see you can create a lot of diversity from starting with simple starting molecules. And what you do is that you take those mixtures and you test them in your cell-based assay, and you see which ones give you an effect that you want. So let's say in this case, the triangles made the neurons grow longer neurites. So now you take this mixture, you have an active compound within it, and you break it down in a clever way into submixtures, such that each one of those submixtures has one thing in common. So in this mock example, the first column, all of these triangles have the red dot at the top. The one next to it, they all have the blue dot at the top, and so on and so forth. And then you test those submixtures again. And then based on where your activity segregates, you can learn just from this one experiment that you want the red dot on the top, the blue dot to the right, and the yellow dot to the left. And then in one go, you can predict that the shape of your active molecule. So instead of having had to test all of these molecules one by one, in a much smaller number of tests, you can go from hundreds of millions of molecules to quickly identifying something that works in your system. So using this clever technique, we were able to screen 400 million different small molecules and identify ones that promote neuroid outgrowth in our assay. So given that these techniques generate tremendous amounts of data, they can't be analyzed in using traditional methods. We make use of machine learning. A lot of the techniques that were mentioned today, such as information theory. We also use support vector machines and random forests and the likes to kind of make sense of this data, but also to integrate it with other types of data with two main goals. One, to discover novel biology, and two, to identify compounds that we could turn into drugs that can promote nerve regeneration. And as an example, from integrating and analyzing the data that we had, we were able to discover a gene or rediscover a gene in a pathway I previously referred to as the extrinsic growth inhibition pathway. I say rediscover because this gene was already known to play a role, but we kind of validated it and showed that it could be a drug target for this pathway. And then we made a completely novel discovery. We found another gene in the other pathway, the intrinsic pathway, that could also be a drug target. And because we had knowledge of both, we were able to identify a single molecule that engages both, and that solved our problem. We now have one molecule that can address both sources of regeneration failure. And we tested this molecule in an animal model of spinal cord injury. So it's represented here, specifically a mouse model. You see the red lines are the axons that are coming down from the brain to the spinal cord. After the injury, they're cut. And they don't grow past the injury. But if you treat these mice with our compound, you see massive amounts of regrowth prior to the injury and also past the injury. And the good news is that this is accompanied by behavioral recovery. So these mice are regaining sensing and they're regaining motor skills. So this is very encouraging. So now that we know, we have all these data from these very cool technologies, we can proceed to developing this molecule into a drug candidate. And this is an iterative process. So we start with this molecule that we know that works in an animal model, and we do iterative steps of improving it, armed with the knowledge of what its mechanistic targets are. So we want to keep improving it so that it doesn't have side effects, but at the same time maintain its potency of binding to the two targets, Rho kinase and S6K. And when we get to the point where we have something that looks safe enough to be tested in humans, but it's also effective in the animal models, we can hopefully go to the FDA and request approval to start testing on humans in a phase 1 clinical trial. So how am I doing on time? Okay, good. I'll stop here. I'll thank everyone in my lab and all my collaborators, and I look forward to your questions later.
Thank you, Dr. Ali. Great talk. Thank you very much. So our next speaker is Dr. Tsoulfas from the Department of Neurological Surgery. Dr. Tsoulfas is an associate professor, and he's going to talk about advances in gene delivery and 3D imaging, offer new insights into supraspinal circuits in the injured spinal cord.
Yeah, I'm going to get in and out a little bit to show some stuff. Okay, the title is quite explicit. I will go to the acknowledgements. This is a collaborative work with Murray Blakemore and Marquette. And here are the different names. Everybody has contributed greatly to this collaborative project, but because of the time is short, I won't go to the specifics, who contributed what. That has been really a great team effort, and it's another illustration these days that collaborative work is the norm because everybody brings different expertise and different aspects to the table because biology has become quite complicated. The problem I'm trying to explain here is in terms of regeneration is the aspect of locomotion. So we know there are different centers of locomotion, both locally at the spinal cord, the local circuitry, but the local circuitry does not work autonomously. It requires supraspinal centers that, depending on the behavior of particular behavior, motor action will ensue. one of the problems with the spinal cord injury and regeneration in general is that everybody has their favorite area to study. And the problem is because it takes really long time to analyze data and look what specific neuron has regenerated and so on. It has become a little bit anecdotal. Everybody uses their own specific nucleus and they say, okay, you know, corticospinal sprouted, and therefore if my behavior changed, then that means it's related to the corticospinal. But motor activity is quite complicated, and unfortunately it's really difficult to pinpoint the nuclei. The neuroanatomy of these supraspinal centers are known for over a century. There is a lot of literature. There is over 50 of them with different subnuclei and so on. So we wanted to actually address this problem, and instead of looking at one specific nucleus, because it's easily accessible or we have antibodies, to look globally, to look at all of them at once. In this way, if there is a particular regenerative strategy, then we can correlate the presence of connections to the site of injury and so on. Here is, there are different locomotor centers. There are all these names: hypothalamus, midbrain, brainstem, red nucleus, motor cortex. And here is more an intraspinal connection, important for step synchronization and speed. How we approach this problem is to use practically combine for different technologies. I will start with the last one on the experiment, and that is the light sheet microscopy. Light sheet microscopy was invented almost more than a century ago by Zygmundi and Siebentrop, and that is really rely on a horizontal light path towards the microscope. These days, there are modern microscopes. Usually the one on the left is the lens and the light path are perpendicular to each other, whether in the light sheet, in this case, ultramicroscopy, the light path, the light sheet is horizontal. That has certain advantages, which I'm going to enumerate for the time, because we don't really, there is only 10 minutes. The next step is tissue clearing. Also, that was more than a century ago. The first person to do was Pantelis in 1911 in Vienna, and took almost 100 years to come with new ways of clearing. And you heard all kinds of techniques lately in the last 10 years, from clarity to disco, all kinds of acronyms. So light sheet, tissue clearing. The next one you really need in this approach is viral delivery. So you want to label supraspinal centers, practically actions that project to the injury. One way to go around this is viral delivery, and we know there are the adeno-associated viruses. Those mostly are anterograde, and they have a particular tropism. But however, you can have what is called capsid evolution. So you can change the capsid, so you can change the tropism, but also the ways in transported. So in this case, we use the AV2 retro. And the fourth part is to have a bright fluorescent protein. There are several out there. Here is just illustrating some of the bright ones at different spectra. In our studies, we have used differently. So by combining these, we can actually reveal in its entirety all the supraspinal centers. And here is a movie. So this is an injection at the cervical level. 4 weeks later, we take this brain, we clear it, we image it with light sheet, and then here is the result. Here is the whole brain. This is the brainstem. This is the cortex. This is the pyramidal tract. Here is the midbrain. Here are some of the cerebellar projections. So practically, we can really reveal most of the supraspinal pathways that project to the spinal cord injury. Therefore, if you have a regeneration strategy, we can see what is spare, not spare, or whatever has regenerated. This is a really nice image, but is mostly qualitative. There are some technical issues that even make it really impressive to look, not really practical to use for regeneration strategies. So we implemented a new protocol, a new pipeline to get quantitative data. Also, what you saw there is you saw a lot of cells in the brain. We know the region by eye, but we don't know the regions of those specific 50 nuclei with their subnuclei. And if it's a particular one that has been preserved or regenerated, will be really difficult to identify and say, oh, you know, this is the cuneate, or this is central amygdala, and so on. Is really hard. So what we do here is after the light sheet, we can render on the images, we can get automatic spot and numbers, but we have to use a different program to get the registration and understand where the nuclei are. So here is a view of a lumbar tracing. Yes, different areas. This is cortex, this hypothalamus, midbrain, and here is the pons. We can quantify these spots, so we know by region how many cells are there or how many cells are left after injury. However, we don't know the nuclei. We don't know exactly what is in these areas. Could be reticular formation, could be all kinds of other names related to the nuclei. Let me get off these and show you what we're doing these days. Here is, this has been traced both at the lumbar and cervical level. And you can see the spots. We know exactly the region, but we don't know the nuclei. And let me take off the green and go back to the cervical. Now, we want to know not only the number, but where these spots are located. And I will go back to the presentation to illustrate the point. So the program we're using is AMAP. It's a validated pipeline to register this data. Remember, the images we're taking is 2D. What you saw rendering is in 3D. So we can take this 2D data and put it in the pipeline. And what the pipeline will look is will register into the software, and the software practically outlines on our brain, that is a brain, this is just background, and each line practically outlines a specific nucleus. Once we have that, then we can register our data. So here is different sections through the brain. The dots are the different neurons, and within each the nuclei. So now we have registered the data. Now we want to get the numbers. This is some other views we have here. So the registration can give us not only the dots and the relative numbers per area, but also we can visualize them with the brain render, the different areas where the spots are. And each color is a specific nucleus. Let me go here, stop this. Here is some of these data that we have gotten now. So here is how we'll look in 2D, the image. The different spots and circles is where the various neurons are. And here on the right, you can export all the data from the 3D on the 2D registration. And here are all the nuclei, most of the nuclei, or the most important we identify. And we can get relative numbers, cervical versus lumbar. We have done these now for all kinds of regions. and injections at the spinal cord. And there are some really interesting, let's say, results, if you can say it. I mean, the literature for the 100 years has been always at a specific area or specific segment of the spinal cord. And, you know, there are quite impressive data, but there has been nothing like we have done now, where we can compare different segments of the spinal cord, lumbar versus cervical, a specific nuclei, what is 40% cervical versus 60% lumbar. For example, take the nucleus of Barrington. Barrington's is involved in micturition. Most of them are really lumbar. And then we can go down to all these areas that people know how are involved with locomotion. So the idea really at this—
Tsoulfas, sorry, I can give you an extra minute.
Okay, so practically the idea is now to take this pipeline and correlate it to the injury or regeneration strategies and practically correlate anatomical position to behavior to quantification. And that is really the ultimate goal. Here, let me show you another brain. This is injury. So now we have done it for injury. Actually, let's go to this. What we want to do now is democratize this process. Here, I'm just showing you our website we're building for each brain in injury. So you can go in and, depending on the region, let's take here this one. This is a gigantocellular nucleus of on the brainstem, and you can actually visualize where the nuclei are, the various neurons are, and then compare injury versus not injury, and then overlay that to behavior. So that's all I have to say for now.
Thank you, Dr. Tsoulfas. Thank you very much. So our next speaker is Dr. Danziger, assistant professor at Florida International University. And he's gonna talk about learning and error in human-machine interfaces and brain-computer interface models. Dr. Danziger, you can go ahead. Thank you very much.
Yeah, thanks for the introduction. A lot of great talks. I'd like to just say, if everything goes well, these are the things that I'm going to be telling you. The first thing is why intracortical brain-computer interfaces need a better model to study. And this was described at least a little bit in the previous talks when we were discussing how invasive these electrodes can be and how we need a different approach. The second thing is to talk about a model we have of brain-computer interfaces that is a bit abstract. And number three is to show the results that we've gotten from this model and how they can be applied to brain-computer interface problems. And then finally, introduce the direction that we're taking this next and our really interesting, more faithful models to brain-computer interfaces. So in general, this is what a brain-computer interface, an invasive brain-computer interface looks like. And so you have a subject, a human or monkey subject. From subject, through invasive recording electrodes, you acquire their neural data, typically from motor cortex. That data is fed through a decoding algorithm that interprets those neural activity patterns. And from that, it controls some external device, maybe like a computer cursor. And then the subject sees what's going on with their device, okay? But the problem is nobody wants to be a subject for these experiments, right? Because you have to implant electrodes directly into somebody's brain. So understandably, for a very experimental technology, you only get a few subjects and those that already have extreme paralysis. So what typically happens is in brain-computer interface studies, you get only 1 or 2 subjects. This is the typical case. And so this is not enough to make rigorous analyses and optimize these decoding algorithms that we need to translate between the neural data we record and the control of somebody's computer cursor or power wheelchair. So the typical approach is to swap out a human subject with a monkey subject because you don't need their consent. But that doesn't solve the problem of not having enough experimental subjects, because it's still very difficult and expensive and time consuming to run a brain-computer interface with a non-human primate. The next approach is to replace the subject with some already recorded neural data set. So the problem with this is that the whole system becomes open loop, and there's a critical connection here between what the subject sees and how they respond. In fact, a decoding algorithm in brain-computer interfaces, what that decoding algorithm is doing is not simply reading the subject's mind. It's dynamically interpreting those neural signals. And so this task of controlling a computer cursor by modulating your own neural signals is more like learning to play a musical instrument than it is sitting back and having your thoughts read. Okay, so this is our approach. It's to keep the subject, the human subject, and use something else for the neural data. Okay, so let's see how we do this. So this is the general idea. What we are doing is, instead of neurons, we are using a human's finger joint angles. This is right here. And so there are a lot of analogies between human finger joint angles and neurons, although I can't enumerate them all. The basic idea is that when you record neurons, there's a lot of signals, just like you have in the hand, and they're in a strange high-dimensional space, right? That is not very intuitive. And what we do is we map these finger joint angle movements to cursor control instead of neural data. And I'm going to show you a video of a human subject using this in real time. And if you've ever seen a video of somebody trying to use their brain to control a computer cursor through an invasive brain-computer interface, you'll notice it looks something like this. I'm sure you will see this as more choppy than it actually is because it's coming through the internet, right? And this is great. So this gives us a platform through which to study something about brain-computer interfaces without needing to put electrodes in subjects' brains. And this work was done with partners at Northwestern University. So let's take a look at one experiment that we've done. Okay, so suppose that a subject makes these 4 hand postures. And this is our decoding algorithm. So our decoding algorithm puts this cursor near target 1 when subject makes hand posture 1. So there's an error. So basically what we would like to do is, for all these hand postures, we know the subject is trying to reach each one of these targets. So let's update the decoder so that now when we pass it through the new decoder, each one of these previous hand postures lands in the targets that we want them to. But the issue is that there are many such decoders that will do this task and do it equally well. So our experiment was to figure out if these different methods produce different results. Right, so on the one hand, we used an analytical solution. That is, basically we just solved a bunch of linear algebra to come up with this new mapping that would create 0 target error. In another, we used an optimization algorithm, least mean squares, to do the exact same thing. Right, so the outcomes of these 2 methods, the green analytical method and the orange optimization method, they have the same practical results. That is, if the subjects make these hand postures again, you now get 0 error. And then we have a third group where we ask subjects to keep practicing, but we don't change their decoder at all. Okay, so what happens? Well, what I'm showing you here is endpoint error. This is mistakes that they made, how close they got to the target. That's on the y-axis. So the higher you are, the more error you're making, and this is normalized to their baseline performance. So lower is better. So here for this optimization, this gradient descent group, we can see that they are performing better and better over these blocks, right? These are blocks of 30 or 40 trials. And we see that the control group, the purple group, also improves their performance, even though they're not getting help from our optimization algorithm that's updating the decoder, this mapping between their their fingers and the target locations. But shockingly, when we use the analytical solution, this green group, not only do they not outperform the control subjects with no help, they don't learn anything. That is, their error stays wherever it was on day one. And in fact, these subjects became so frustrated they would not return for future days. So here's a trajectory example. So for our gradient solution, the orange group, you can see that their trajectory is going between targets. This is a difficult task, so it's not completely straight, but they're making it to the targets. And here's the green group where we have the analytical solution, and their trajectories are basically a mess. So the question is, why? Given that for both this green group and the orange group, if the subjects had just made their same hand postures over again, they would have gotten 0 error. So why is there such a massive difference, right? Why is there such a massive effect between these two groups? And maybe this can teach us something about how we should use brain decoding algorithms in actual brain-computer interface studies. Okay, so our hypothesis was basically that the trajectory between targets was very important. So the gradient solution, this optimization solution, made very straight trajectories in the sense that it changed the decoder only slightly from where it was previously, right? So whatever trajectory the subject had made previously, their trajectory is going to be very similar this time. So here, the lower you are on the y-axis, the smaller the change was made to your decoder. Likewise, the analytical solution, much higher changes. And here's an example of what this would look like. You have an original trajectory. The update with the orange method nudges you closer to the target you want to be at. The green update method also gets you to the center of the target you want to be at, but it takes you through a very unusual path. And we think this is the reason why subjects had so much trouble with it. And it turns out that the current iterations of effective brain-computer interfaces, like refit out of Chenoy's group, rely heavily on the trajectories that subjects are intending to make. So these theoretical results on this model system also agree with what we're finding in practice on brain-computer interface studies. The only difference is we can test 50 subjects and show this with statistical significance, right? As opposed to anecdotes from one subject at a time. So quickly, where are we going next? Well, we are keeping the human subject with their joint angles. But now what we are going to do is try to make a more faithful brain-computer interface model. So these joint angles go into an artificial neural network model of motor cortex. So therefore, when the subject inputs their joint angles, what this generative model outputs are plausible artificial neural firing rates from motor cortex. And then what we can use are actual decoding algorithms that are being used in the field right now. And we can test those actual decoding algorithms with statistically significant samples with a human subject in a loop. So this gives us the best of everything. And the only concession that we make is that we are using a model of motor cortex, a deep neural network model, instead of the real thing. So here is our basic pipeline, where what we're doing with our help from our collaborators in Northwestern University and EPFL is we track the monkey's hand as a non-human primate makes reaches. At the same time, we gather neural activity from their motor cortex, and then we use these data and the hand positions to train our intermediate neural network, our generative model. And I see I'm running out of time, so I'll just say, as soon as this is done, as soon as this network is trained on this data set, we can then have a subject sit in the chair. We plug the subject's hands into this recurrent neural network, which is a model of motor cortex, and those signals go right to a decoding algorithm. So a human being can, a healthy, intact human being can non-invasively interact with state-of-the-art decoding algorithms. And lastly, these trajectories show an actual brain-computer interface, and our model, you can see they're very similar. So we have done experimental validations of this. Thanks so much for your time.
Thank you very much, Dr. Danziger. So our last speaker is Dr. Cabrera, an associate professor at the Department of Ophthalmology and at the University of Miami. And she will talk about investigating predictive models for early diagnosis of cognitive impairment using multimodal eye biomarkers. So, Dr. Cabrera, you can go ahead.
Good morning, everyone. It is my great pleasure to speak here today. I'm going to present results from a study that investigated predictive models for earlier diagnosis of cognitive impairment using multimodal eye biomarkers. Firstly, I would like to thank to all of our collaborators and research participants involved in this study. And here are my disclosures. Okay, here is the outline of our talk. I'm going to show you the methodology and the results that we have obtained after investigating whether fractal complexity and lacunarity analysis performed in sectoral regions of the retina reveal or not alteration in patients with cognitive impairment that may be masked in the analysis when considering the whole retinal branching pattern. We also investigated how the microvascular network complexity and neural function alterations of the retina contribute to differences in cognitive function. As we know, the projected growth of the worldwide elderly population guarantees a growing epidemic of dementia. Aside from finding a cure, the holy grail of dementia-causing diseases research is the discovery of biomarkers. However, the critical barriers to primary prevention of cognitive decline in dementia-causing diseases are the lack of rapid, non-invasive, objective, sensitive, and low-cost biomarkers. The eye is the only organ where one can examine directly the blood vessels that are part of the cardiovascular and the neurological system, because the retina can indirectly reflect the microenvironment of the central nervous system. Retinal imaging and visual performance features, as well as retinal circulation, could be investigated and used to see the brain through the eye. A major advantage compared to neuroimaging techniques is that imaging the retina is non-invasive and can be performed at low cost. Our approach takes advantage of multimodal techniques that are user-friendly and low cost. These techniques can give us information about the structural, vascular, and functional mechanisms of the retina. Our objective is to investigate the correlation between the retinal vascular complexity and neurodegenerative changes in patients with cognitive impairment to find whether evaluating visual capacities in elderly individuals may add predictive value of early visual pathway injury associated with cognitive impairment, and also whether novel multimodal eye biomarkers for early detection of cognitive impairment in Alzheimer's disease or other dementia-causing diseases could be introduced at a low cost. Okay, let me show you the methodology that we followed. Prospective subjects with cognitive impairment were identified in a non-systematic fashion as they appear in the clinic or identified from a population attending adult care centers and community clinics. Exclusion criteria were age under 55 years and the presence of any ophthalmic history before recruitment. Also, patients were excluded if they have other neurodegenerative pathologies. Cognitive function assessment was obtained with the MOCA test. We also conducted full field electroretinogram, color vision test, and use advanced retinal imaging. Optic disc and macular center images were scrutinized for pathology, and subjects without any ocular history except for cataract surgery were included in the analysis. Smoking history, diabetes, hypertension, and cardiovascular disease were considered comorbid medical conditions related to retinal vascular alterations. We also conducted several statistical analyses to find statistical significant differences, if any, between the groups. So our image analysis took into account retinal vascular parameters such as the artery-vein ratio, asymmetry factors, and diameter of the vessels that were calculated with the commercial software. Once the vessels were segmented, the corresponding skeletons were obtained and used in the fractal analysis. Okay, here we had the results obtained after analyzing the regional retinal branching pattern. Out of 69 participants, 32 had cognitive impairment. After applying all exclusion criteria, A total of 20 subjects with cognitive impairment and 19 cognitively healthy controls were included in the final analysis. We found functional loss in color vision, smaller electroretinogram amplitudes, and larger peak times or implicit times in the subject with cognitive impairment. Also, the multifractal dimension was lower in individuals with cognitive impairment, and we also noticed drusen-like regions in the peripheral retina along with pigment dispersion. And let me show you in the next slide so you can observe better. You see, you notice drusen-like regions in the peripheral retina, most of them in the superior quadrant, along with pigment dispersion in patients with mild and more severe cognitive impairment. The red arrows indicate the location of the drusen, and white spots observed are extramacular locations. These areas enclosed by the orange rectangles indicate the locations where pigment dispersion was observed. Two earlier studies that may add to this evidence have described the presence of amyloid beta in retinal drusen deposits. Also, abundant amyloid beta pathology has been detected in patients with Alzheimer's disease, mostly in the periphery of the superior quadrant. Well, we found significant alterations in the microvascular network in individuals with cognitive impairment, and this was actually a result that was revealed in both the regional and the whole retinal branching pattern analysis. We also found that the multifractal parameters were significantly greater in the optic disc region in cognitively healthy individuals, and also that the effect size for the optic disc region was greater when compared with the whole retinal branching pattern. Also, the lacunarity values were significantly greater for participants with cognitive impairment than controls in the optic disc region, but significant differences between these two groups were absent in the whole retinal branching pattern and macular region. And you can see in table 1, we have very limited time, so more detailed information could be found in our paper. In addition, we found that the multifractal parameters were significantly associated with the MoCA scores and implicit time in the macular region, but these associations were absent in the optic disc region. Also, we found a significant negative correlation between the lacunarity with the implicit time parameter, and this was only observed when considering the whole retinal branching pattern, and also correlations were obtained between the generalized dimensions and the implicit time parameters, and they were higher in the macular region. These are the results we obtained for the ROC analysis, which was conducted for the region that had a greater effect size, and as you notice from the previous result, was actually the optic disc region. This analysis shows the advantage of the multimodal diagnostic biomarker approach based on the retinal structure function relationship. You can see that the model using the multispectral parameters along with the lacunarity and the implicit time was the one showing the best performance and the highest area under the ROC. Okay, well, here are our conclusions. This pilot study introduced a more efficient methodology to characterize the retinal vascular network by combining multifractal analysis, multifractal spectrum analysis, and computing the lacunarity parameter. Our initial analysis, as I already showed you, suggested that our study's small sample size may have accounted for masked alterations in patients with cognitive impairment when considering the whole retinal branching pattern. However, the sectoral region analysis showed that the sample size did not affect the significant difference and large effect size found for the retinal vascular fractal dimension parameters, lacunarity, and multifractal spectra values between the two groups and the associations between the retinal vascular and functional parameters. Also, our sectoral region analysis revealed better the alterations in the multifractal analysis parameters for individual retinal areas and their significant correlations with flicker implicit time. Instead of individual biomarkers, the investigation of combined multivariate vascular parameters in the retina sectoral regions might provide a useful clinical marker of cognitive impairment. In addition, our results support the use of a multimodal diagnostic biomarker approach based on the retinal structure-function relationship, and also has the advantage of a low-cost implementation in community settings. I think that the full extent of our approach clinical applicability is provocative and still to be determined by using a larger and more diverse sample size with longitudinal study design. But overall, this pilot study would promote the investigation of multimodal longitudinal models using the eye as a window to the brain to facilitate the low-cost approach for cognitive impairment detection and also change our understanding of cognitive impairment. With all that said, I would like to thank you for your attention.
Thank you very much, Dr. Cabrera. Thank you to all our speakers for participating today. We have few questions, so I can start to read the first one. It's from Dr. Barro, and I think it's for Dr. Khizroev. So did the magnetoelectric nanoparticles cause localized heating of the neurons after being stimulated?
Yeah, 1 second, let me do video. Oh man, oh yeah, okay. Thank you. So the question is whether the particles heat. Oh yeah, that's a very good question, by the way. That's DARPA guy, our neuroscientists always made us actually do a study. No, they don't, because they're very efficient. We control their physical properties, including that energy emitted by them. Remember, I showed the hysteresis loop. Hysteresis loop tells you how much energy you will lose if you're not careful. That's in our control. We make sure all the energy we provide gives, the energy we give is magnetic. The energy we use when we stimulate is electric. So because of quantum mechanical relationship between these two, we can make sure our yield right now is almost 90-something%. So barely anything goes to any other energy with the exception of electric. So, and of course, heating, it depends on time and so on. I'll tell you exactly more specifically. We conducted experiments for several hours. There was not slightest detectable temperature increase. Probably because it's extremely local as well. Who knows, that's why. Thank you.
Thank you. So the next question is for Dr. Danziger from Prasad. Dr. Prasad, great talk. Thanks for presenting your work. I was wondering, how would you get joint angles as reliable control signals for a spinal cord injury person who has spasticity and minimal movements in their hands? Thank you.
Yeah, that's an interesting question. Thanks for that. The experiments that we do with our brain-computer interface model are on healthy, intact subjects. So what we're doing is using their fine and dexterous control of their fingers to sort of drive our models and produce our faux neural activity. But what you're asking is sort of turning this into a therapeutic device rather than a model, which is really cool. We've done some work on that, especially my collaborators in Northwestern, where instead of using a data glove to capture their finger movements, what you do is put accelerometers on their shoulders, or you have them wear a shirt that can capture shoulder and trunk movements. And then what you do is map all the residual movements that those patients have into the control of their assistive device, like a power wheelchair. So Musa Avaldi and the Casaglio in Northwestern University of Genoa, those are great professors to check out who've been doing work there, and I've contributed a little bit to that. But I think that's a really interesting way to take this work.
Thank you. So next question, I think is again for Dr. Danziger from Alcacrom, Dr. Alcacrom. Great talk. Can you augment the model performance with using surface EEG data in addition to joints position in humans?
Yeah, that is that's a recommendation that we've had a number of times. So I think you're in good company in thinking along those lines. So the idea is that in addition to all the high dimensional movements that we get in our fingers, we could also do EMG, and that would give us even higher dimensionality of control and mimic the cortex even closer. There are some technical implementation details. For example, our models have to run very fast because this all has to be in real time and closed loop. So we would have to get very clean muscle signals, and the degree to which the muscle signals are independent of the finger movements is also important. So we can continue to add new dimensions, but they're only relevant from a decoder's perspective so long as they're not highly correlated, right? As long as their mutual information is low. So yeah, insofar as we can pick the right muscles and get the recordings and signal processing fast, yes, we can expand, and we're looking for more signals, and that might be a great way to go. Thanks.
Thank you very much. So the next question again is for Dr. Danziger from Dr. Dakistra. What are some of the hypothesized reasons why the best screen paths from one target to another are so indirect? What does that say about the nature of corresponding brain states and trajectories through them?
Oh my God. Okay, I'm going to have to get everyone here to our lab meetings because these questions are so incisive. So actually, I don't think that the most efficient paths between these targets are these curved trajectories. What I think instead is that it is just difficult to control a computer with a brain-computer interface or our BCI models. And I think that's why you see these unusual trajectories. But people do try to move in relatively straight lines when you show them a computer cursor. In fact, we showed this in a J Neuroscience paper in 2012 where we were working on this model. And what we found was people internalize the geometries of the devices that we showed to them. So meaning if we show them a point on a plane, they infer Euclidean geometry and do their best to move in straight lines. But if we show them like a simple robotic arm, the configuration space of which is a torus, they make nonlinear movements between targets, non-straight line movements between targets that correspond with the configuration space of that robot. So I think there's a lot of room to manipulate through visual feedback how people move between targets, and it's a great knob that we can use to improve people's performance in brain-computer interfaces. But for cursors, I expect people really are trying to move in straight lines over the screen. Thanks.
Thank you. Regarding our previous questions, Dr. Alcacrum said, meant to say EMG and not invasive EEG.
So sorry, what was that? He meant to say?
So there was a question, I forgot the question. Let me get to the question. So before he asked, can you augment the model performance with using surface EEG data in addition to joints position in humans? But he meant EMG, not invasive EEG.
Ah, great. Actually, I assumed he meant EMG, which is how I answered the question. So we're on the same page, right? That answer was with reference to EMG, myography. With EEG, those signals would be too slow for the speeds at which typical intracortical brain-computer interfaces operate, where you bin neural activity in 25 to 200 milliseconds. But don't tell Jose Principe I said that because he was railing against binning neural data earlier.
Thank you very much. So next question is for Dr. Barro from Dr. Bio. Have you studied the effects of single residue mutation of the residue that are not part of the voltage sensor domain of the KCNQ channel?
Yeah, the answer to that question is yes. I studied many other mutations out of the voltage sensor, even out of the voltage sensing domain, towards the gate even in the S4, S5 and S6 port and the gate. But in casing Q1, so the one that it's expressed in the heart. For casing Q2 and casing Q3, the one that are expressed in neurons, it's a little bit more challenging. And the exciting part of it is that there is no publication whatsoever until today about how the voltage sensor move at all, for example in casing Q2. So the few publications targeting voltage sensing or rearrangement of the voltage sensor using voltage clamp fluorometry come from few labs in the world and only tackles mutations within the Volta sensor. So for KCNQ2, there is not even any single publication. So there is no clue about how this channel really works. So what I'm trying to say is that we are putting a lot of effort to just make this technique happen with this channel. For some reason, this channel has been really hard to deal with. And the few mutations that we are doing seems to be working within the Volta sensor. This is our primary goal at this point. But of course, we are planning to, extrapolate it to the gate and understand how the gate actually works in conjunction to the Volta sensing domain. Yeah.
Thank you very much, Dr. Barro.
Yeah.
The next question is for the Danziger. Again, it's a long one, so you probably have to read it again. So how accurate will be the approach of combining both joint angles and stimulate a brain network of the motor cortex in the case of motor system disorders? For instance, in spinal cord injury, there will be a strong imbalance between the precision of the movement, poor, and the motor excitability, high. Would such approach be able to decode such imbalance?
Yeah, so this is a fair point. And, I don't want to oversell exactly what our models are capable of, right? So I'm not telling you that I can model motor cortex full stop, right? Actually, what we're doing is modeling motor cortex in a very narrow range of its operation that is maximally relevant to brain-computer interface tasks. So we don't study the monkey's behavior over its entire lifespan doing all sorts of activities. It's very narrowly focused on fine digit manipulation during reach and grasp tasks. So when I transfer that to my brain-computer interface models where our human participants are doing these kinds of things with their fingers, the claim is that it is a good model of motor cortex in that specific domain, right? So as soon as you start talking about things like spinal cord injury, where the general excitability of neural pathways has changed, or in the aging brain, I kind of right now have to throw up my hands there, because the quality of our models are limited by the data sets that we have, and we don't have data sets to train models to do those things. So right now, no, we can't do that, although that would be a really great extension of this project. It's pretty expensive to get that data, and our Northwestern collaborators have been very patient in setting up those studies. So we're going to walk before we can run, but thanks, good question.
Thank you, Dr. Danziger. Regarding the comments of Dr. Alkagrom, Dr. Dietrich, would you like to say something? Because he says that you would like to answer this question live.
I sent people various questions. Who are we referring to?
It's so meant to say EMG and non-invasive EEG.
No, that was not. No, that was not me. Sorry.
Okay.
Yeah. I did want to ask Dr. While I'm on the call, I would like to ask Dr. Barro a question about the neuronal channels. Post-traumatic epilepsy is an extremely important problem in patients, and I was just wondering, number one, what is the response of these channels to brain trauma? And number two, could the mutations have any effect on increasing or decreasing a patient's vulnerability to the PTE?
Yeah, very interesting question. I was actually answering to your question by email. So the details, you will get it by email. Yeah? But in principle, it's little known about it. So nobody knows really the contribution of this particular channel to TBI, and nor even less it's known about patients carrying inherited mutations, the susceptibility to TBI. So I started a couple of years ago testing myself samples from patients suffering TBI in a collaboration with Dr. Keane in the Physical and Biophysics, and the results were at that time inconclusive, but we saw that the trend of these samples, people were receiving shot, gunshot, and then they were extracting blood samples and plasma, and there were some particular samples that actually inhibited one of these channels, the wild type of one of these channels. So there might be a correlation between, so my hypothesis, my idea is that there is something, some molecules in this patient or something after a TBI might really be released onto neurons and might inhibit not the expression of the channel, but the function of the channel. And that in turn causes hyperexcitability, in these neurons. So now, but this is not published or anything is known about it. Now, with the mutations, it's a little bit more complicated because you have gain of function mutations in these channels and loss of function mutation in these channels, with both causes the same phenotype. They causes epilepsy, so hyperexcitability. And this is completely, the mechanism behind it is completely unknown. So this is really a puzzle. For the loss of function mutation, it's easy to imagine that would cause, silencing of the, hyperexcitability, because you have more or less potassium secretion out of the cell. But with the gain of function, it's a little bit trickier. So there's no known. So people think more in as a network process there with the gain of function mutation. So to your question, TBI, it's really interesting, and I think, yeah. So we should explore that for sure.
Thank you very much.
Thank you.
Can I ask a question to Dr. Barro?
Yes.
Sure. Is this channel expressed, where is expressed in all neurons, specific neurons, and how can you correlate these epileptic attacks to where it's expressed?
Yeah, no, this is a very interesting question. And again, it is currently under, dispute. So people, the problem is that the antibody for KCNQ2 and 3, particularly KCNQ2, are really bad. So people don't know really where they are. And then there've been other methods to just map where these channels are expressed. So it looks that they are almost everywhere. So for sure, pyramidal neurons, they are expressed. Yeah. But then the question is whether they are more expressed in in excitatory versus inhibitory neurons, and whether there is a segregation when you have loss of function and gain of function mutations. This is not really clear what exactly is. What it is clear in the neurons that people know the channel is expressed, it is clear where. So it is in the initial axonal segment, in the synapse, and in the soma. And it is heavily in the IIS, so in the initial axonal segment, and that's why people believe this is a channel important for this, breaking or functioning as a break of repetitive neuronal firing. But more than this, also, what's the neuron specificity? It's really ambiguous.
Yeah. So you said layer 5 of so if I understand, it's the same one that is expressed on the hilus. The initial segment is called hilus. So that hilus can change. And so, for example, after spinal cord injury, is known that this hilus can be reduced in size, and all the potassium channels, I don't know if it's the same, are around that hilus. And that has been correlated to particular functional states or disease.
Yeah, well, this is interesting. These channels are also expressed in the peripheral nervous systems. It's involved in pain, for example, too. And yeah, so but it's interesting what you just said. Yeah.
Okay, there's one more question from Noga to Pantelis.
I read it, yes.
Okay, all right.
So how can we use this imaging method to target neurons higher up in the connectome of different motor systems that do not project directly to the spinal cord? My answer is yes, you can use them anywhere you want. So since are retrogradely transported, so you need to hit the target of that particular projecting nucleus you want to see. So can be used anywhere, depends on your, how good your injection is. yeah, can be used everywhere, even, I guess, in the peripheral nervous system.
Thank you. So are there other questions?
I could ask one to Sakharat, if that's okay.
Sure.
Can we, what's the spatial specificity that we can get those microbeads into regions of the brain? So suppose we want a non-invasive brain-computer interface, but we need the beads in motor cortex and premotor cortex. Do we have delivery capabilities like this?
You call microbeads, we call them nanoparticles, right? Yeah.
So, yes, I didn't mean to insult the spatial scale of your devices. Yes, nano is much more impressive.
There are two basic ways to deliver particles, to deliver stimulation in this case. You either physically move particles to the destination, and actually it's not a simple technology. It's called metastable physics delivery, and it took us a couple of years to build that machine. I have a video to show you how we can do it with this precision of 50 micrometers. We can do it. But the challenge is that delivery strongly depends on the size of nanoparticles. If your nanoparticles have distribution, so some of them will go there, some of them will get diffused somewhere else, and you will lose some of them. So better approach, we realized what I showed today was we localized field profiles. So we just drop particles. We actually did this experiment in animals as well, but we were more careful with this one. We wanted to show reproducibility in some cell cultures. So we just drop micrograms of particles in a Petri dish, regular size, centimeter diameter, so. And then what we did, we used our field sources to localize particles, what I was showing you. No, not localized stimulation. Particles could be everywhere, that's the thing. So you have to make sure they're not going to ruin anything, but so concentrations are so small, I would say approximately 10 nanoparticles per neuron. They're everywhere, but only the ones you want to be activated are activated by application of special field profiles. That's what we did in that experiment. Their resolution, I showed you, about 80 micrometers. So, so we can put sources outside the head, and with that precision, extremely difficult to make. I skipped all the engineering. We worked with a very powerful company in Irvine. They actually built it for us. So that was a DARPA project.
I see. Yeah. So because of your targeting is so specific, it doesn't really matter that you have to where you need to move the beads to. They're already there. You just
Exactly.
That's cool.
But by controlling fields, it's all done with wireless fields. Yeah, exactly. So.
Other questions? No. So we can conclude the first day of the symposium. I would like to thank all the speakers again. Great talks and great discussions. And I will thank you everybody for joining us today. And as a reminder, tomorrow there will be the second part of the symposium. We will have 3 other sessions. One is the brain-machine interfaces. The second one is the aging brain. And we will have the live poster presentation. So please join us tomorrow morning at 9. And with that, I don't know if Dr. Rajguru or Dr. Dietrich would like to add something else.
I'll just add again, thank you to all the speakers as well as all the participants. Thank you for joining us today, and we'll see you again tomorrow morning. The box link that has been shared with you has all the posters, videos, as well as the poster PDFs or PowerPoints that everybody has posted. And you should have viewing privileges, and you can already start seeing them. And then tomorrow we'll have 10 live poster presentations and an award ceremony. Thank you. We'll see you tomorrow.